Okay, so welcome today and thanks for joining to our second part of the Nuvisan live talk, how to translate your oncology target into a preclinical candidate. So Martin and I, we are very happy to have a second part on this Nuvisan live talk. And since we have a huge interest in our first talk, we would suggest to wait additional 2 minutes to give also all the other potential interested people the opportunity to join. But in between, we have a poll, which is very important for our marketing guys, and this should come up now. And yeah, we would like to answer, if you like, if you have ever worked already with Nuvisan before. And yeah, then we wait additional 2 minutes, and then we will start. Sounds good. Good, should come up now. Hopefully this works. And we are very interested to see if we maybe know already. Last time, I think we had certain overlaps from our previous work history as well.
Yeah, so that was nice to see some familiar faces, at least virtually.
Exactly. So I think, let's wait maybe for additional seconds. Yeah, I think we should be completed now. So thank you for your patience. And yeah, let's start. So I shortly like to introduce myself. So we cannot click forward here.
Yeah, I think we can start anyway with it.
Yeah, I think. Yeah. So I'd like to introduce myself. So if you haven't joined our first talk yet, so I'm Dr. Carlos Stresemeyer. I'm heading the therapeutic oncology research department in the Innovation Campus Berlin, which is part of the Nuvisan group. And basically, I've joined industry 13 years ago as a scientist at Bayer. And during this time, I was supporting numerous projects from target validation up to IND preparation. And currently, my group is focusing on all the core oncology activities, meaning more or less in vitro as well as in vivo characterization of compounds, evaluation of biology, and also diseases which might be relevant for oncology research. And I would like to give my colleague, Dr. Martin Lange, also the opportunity to introduce.
Thanks, Carlos. So it's a pleasure to join the session again here. Welcome also from my side. My name is Martin Lange, and I'm head of the translational research group within pharmacology at ICB. I'm a biologist by training with a background in oncology drug discovery, mainly through the time that I spent at Bayer Pharma as a lab head and project leads in lead optimization projects in oncology, focusing on translational aspects of these projects, moving into preclinical and clinical development. And that's basically also the role that I have here now at ICB. And yeah, I look forward to today's presentation once more. And let's go to the house rules here, I think is coming up next.
So next slide, please. Yeah. Very quickly, some house rules. So your microphone is muted by default today. You have a Q&A chat box. So for your burning questions, so just type in potential questions which might arise during the talk. We will have a Q&A session and we can pick up those questions at the very end or even afterwards. In addition to that, this talk will be recorded and slides as well as recording will be sent to your inbox. So next, okay. So yeah, in the preparation round for Martin and I, we were thinking, okay, so we cannot just simply present the same stuff again. And having now the opportunity of having a second part, we were focusing a little bit on the previous poll, which we did in our first talk. And the question was, what are your main challenges in oncology and drug discovery? And as you can see here in the chart is that a lot of those questions or answers were circling around target identification, validation, mode of action characterization, as well as some translational aspects, as well as selecting also disease models. And therefore, we were designing a complete new talk, more or less starting again with a short introductory part and also describing potential services which might be of interest to you. But then we would like to focus on just one real life example, starting more or less from the very beginning of a screening campaign, in this case, a phenotypic screening, talking then about how we are doing target deconvolution, how we deepen the mode of action understanding of our new lead series, up to in vivo profiling at the end. And as promised, yeah, we will have a Q&A session at the end. Okay, so Martin, I think that's your part.
Yeah, thanks. So yeah, before we come to this real life example, we briefly want to introduce Nuvisan and our capabilities around drug discovery and development. So we are a fully integrated science CRO, which means that we can really offer the complete package all the way from drug discovery into the clinic and clinical development. Here at the Innovation Campus in Berlin, we are primarily focused on the discovery and non-clinical stage. And Carlo will talk about that in a bit more detail later. In addition to the Innovation Campus, our other capabilities are nicely complementary to that, focusing more on the non-clinical and clinical aspects. So we can offer services around DMPK and bioanalysis, as well as the complete package of clinical development and data management. And finally, services around API synthesis, formulation development, pharmaceutical services, and clinical trial supplies. So that's really unique, I think, that we can offer the complete value chain and be active in all these phases together with you as a customer. And so, yeah, as mentioned, let's go a little bit deeper into the Innovation Campus Berlin. And on this image, you can actually see the building that Carlo and myself are currently in. We are on the second floor, I think. And this is actually the building that most of the ICB colleagues are in. So we are really working under one roof. The medicinal chemistry colleagues are somewhat on the side in the building to the right, so they have their specialized labs there. But you can already see from this picture that we are really in close proximity to each other, which really allows for a very direct interaction. And if you're reasonably fit, you can see every colleague in basically 5 to 10 minutes by foot. So that's a really good advantage here. Okay, so what is the Innovation Campus Berlin in a nutshell? We can offer drug discovery teams with really extensive expertise in pharma R&D through the time that we spend at Bayer Pharma and other companies. We have fully integrated research functions that cover the complete drug discovery value chain. And we have extensive expertise in a number of indications with a strong focus on oncology. Moreover, there is also a strong footprint in small molecules, and we do have access to Bayer's high-quality compound library. Our infrastructure is fit for purpose for drug discovery, and we have, I think, really nice state-of-the-art equipment here.
Yeah.
And last but not least, of course, our leadership team also has quite extensive industry experience based on the time at Bayer. Okay, so that's the ICB in a nutshell, and yeah, Carlo will go into a little bit more detail on how we do drug discovery.
Yeah, thank you for the global framework of Nuvisan Group as well as Innovation Campus. So I like to spotlight a little bit more what's the ballpark where the Innovation Campus Berlin is really working in. And as you can see, and as it has been mentioned already, so we are basically able to serve the full value chain from target identification up to preclinical IND enabling. So what does it mean? So for example, we are capable of to translate potential basic research data into a real target identification campaign and also a target validation campaign later on, which might lead then also in our organization to a screening campaign leading to certain hits, which we are capable of optimizing up to real clinical candidates and leading to a preclinical IND enabling characterization. And that's one potential scenario, but there are also different entry points in working together with us. For example, if you would have already established lead series or compound, you can also come to us. We can pick up this compound. We can deepen the mechanistic understanding of your series and further optimize it to a real candidate. So there are different opportunities of working together with us. And something which is really necessary for doing this in a very efficient way is that basically we are really working hands to hands together in a cross-functional team. So yes, we have different departments being focused on HTS or chemistry, or we from therapeutic research, as well as preclinical compound profiling, dealing with DMPK, tox and safety issues. But we are basically forming a cross-functional team around your project, and we are working together because there are a lot of overlap areas where you need a cross-functional team to really tackle all those potential questions which might arise. And I think that's one of our basic strengths which we have at Innovation Campus Berlin. Talking now a little bit more about Innovation Campus pharmacology capabilities. Here on this slide. So first of all, we can bring a lot of expertise in biologic as well as a small molecule. We have a biomarker discovery group, which can assist you in assay development as well as in validation along the whole value chain. We are being supported in all our activities, having a bioinformatics group. And as mentioned already, yes, we can support you with IND-ready reports and the preparation of it. Coming more to the in vitro activities, we have a functional genomics and expression platform using different CRISPR techniques for different purposes, even the screening approaches. We can do bulk and single cell next generation sequencing, and actually we are also a service provider for 10x genomics. We have a FACS histology and microscopy unit. We are very specialized in developing customized cell-based assays for your specific targets. So we did this very often, and people come up with new targets and we can really think about what to develop. And we are working in our models with primary cells, cell lines, 3D models, as well as IPCs. In the in vivo part, we can offer xenogenic as syngeneic tumor models, doing a lot of different anti-tumor efficacy studies, even also especially in the immuno-oncology field. We can offer PK/PD studies, which are often also connected to the biomarker questions. We can offer metastasis models, and we also have radiation and imaging capabilities. And you might have heard another, or you might have attended another LinkedIn Live talk from our colleagues Charlotte Kopitz and Eki Mai. So we have a strong footprint in oncology, but we are also capable of offering different in vivo models, especially in a lot of different other indications. And so just reach out to us if you're interested in this topic. Okay, so that leads me to our project example, and that's Martin's topic of his heart, I would say.
Yeah, that's definitely true. So yeah, the topic that we brought for you today is one project example of a project that was carried out at Bayer with actually many colleagues now having transferred to Nuvisan, some colleagues still working at Bayer. And it's about targeting YAP1/TAS in cancer. And in case you're not familiar with these two proteins, so we consider these two proteins, which are highly related, for sake of simplicity, to be the same for this talk. And to give you some background to this and why these proteins could be of interest for cancer therapy, actually it comes from early work in a model organism, the fruit fly Drosophila. So, and images of these flies you can see on the screen here, in case you're not a biologist and you did not recognize directly. And what these researchers found is that if you overactivate the autolog of YAP1/TAS in a fly, what happens is, what you see on the right side is the tissues of the fly present with massive overgrowth. So basically it was found that YAP1/TAS are important to tell organs when to start growing and when to stop growing. And as is very obvious from this fly on the right side, if you overactivate these proteins, it leads to massive tissue overgrowth that almost looks like a tumor already. And in fact, because of this, it's easy to imagine that if these proteins become deregulated, it may induce cancer. And that is in fact the case. So as of now, there's quite a bit of evidence that there's a strong role for YAP1/TAS signaling in cancer, not only in flies, but also in model organisms such as the mouse, and unfortunately also in humans. So what are YAP1/TAS? They're effector proteins of several signaling pathways. If you look at this complex cartoon on the right side, you see that YAP1/TAS are proteins at the nexus of several signaling pathways. So they integrate various signaling pathways, and they interact with these transcription factors called TEAT transcription factors. And that leads to an activation of target genes. And this is where the oncogenic properties lie, because these target genes encode genes important for increased cell proliferation, resistance to apoptosis, and induction of cell migration. So that's the oncogenic program that is being activated by YAP1/TAS TEAT. They are typical oncogenes, which means that if you overexpress them in a model system, they induce cancer. And in fact, of clinical relevance, they are